Thank you very much. Thank you very much, all of you, for coming. Okay. There is a balance between cold and hot tumors, and most tumors exist in a cold tumor microenvironment. This kind of tumor microenvironment consists of an intratumoral immunosuppressive environment, and it consists of a variety of cytokine, chemokines, and trophic factor, such as TGF-beta, IL-1, IL-4, IL-10, and VEGF, and a variety of other immune cell population. On the other hand, there's a hot tumor microenvironment, which consists of these chemokine and cytokine, such as IL-1, IL-2, or TNF-alpha, as well as damage-associated molecular protein. These factors recruit the immune cell population, immune cells such as cytotoxic T cells or NK cells, to induce inflammation and immunogenic response. Our question is, can pulse ultrasound, FUS, shift the balance between the cold to the hot tumor microenvironment, from an immunosuppressive environment to anti-tumor immunity? And for that, we wanted to ask a question: what are the transcriptonic and genomic changes that occur after we treat the tumors with FUS? For that, we first investigated the natural history growth of cold tumors, which means that we didn't give them any treatment. We injected both types of cell lines, 41 or B16 melanoma or breast cancer, into the mice flank, and when they reached the size of 5 millimeter in diameter, we defined that as day 1, and from that day on, on every other day, we harvested the tumors and characterized their proteomics profile. As you can see here, the heat map that's shown here is the proteomic profile, which was normalized to day 1. We also measured the tumor size, which were kept on growing throughout the whole experiment. Another thing that we noticed that we saw a decrease in anti-tumor immunity factors, such as IL-1 or IL-10 or TNF-alpha, and an increase in intratumoral immunosuppressor, such as IL-4 or VEGF. In 41 breast cancer, we saw different behavior, but overall the same pattern. We saw that the tumor kept on growing throughout the whole experiment, and we also saw an increase in intratumor immunity factors such as IL-1, a decrease such as IL-2 or TNF-alpha, and an increase in intratumoral immunosuppressive factors such as VEGF that went down. Then we moved to sonicating, to treating these cells, these tumors, with FUS at 6 megapascal. Why 6? If you're wondering why 6 megapascal, you should wait to our next talk, which she would explain that, Dr. Peru, my colleague. Anyway, we used the same model, and on day 1, we treated the tumors with FUS at 6 megapascals, and at the period of 72 hours, we harvested the tumors and characterized their proteomics profile. As we could see here, in the period time of 72 hours, we got a significantly smaller size of the tumors compared to the control. Another thing that we noticed was that we got a clear inflammation response at the first 24 hours after treatment with FUS. A thing to mention that I would like to point out is TNF-alpha, which went up, and VEGF that went down in its expression. For 41, as I said before, it was different behavior, but overall the same pattern. Overall, the tumor size was smaller in the period of this experiment, and an increased inflammatory response at the first 24 hours after treatment. Then in another set of experiments, we wanted to understand or to investigate what are the changes, the genetic changes that occur after FUS treatment. We did that by RNA-seq analysis, and you can see here we found in this volcanic plot, we saw many dozens and dozens of pathways that were overexpressed in red, or did their expression was decreased in green. A thing that was that we noticed that if we took the top RNA-seq pathways, we saw that most of them were associated with anti-tumor immunity response. To summarize, tumor microenvironment varies according to tumor type and the time of growing. We managed to see that the tumor growth was reduced by treating the tumors with PFAS. And we got a very clear inflammation response at the first 24 hours after PFAS treatment, and RNA-seq pathways were associated to anti-tumor immunity response. And with that, don't have a lot of time, so I'll just say thank you, and thank you very much for Joe Frank's lab, Dr. Frank, and Dr. Scott Brook.
Thank you so much. So we have time for a few questions. Well, so I have one question. Do you have thoughts about the time points are hard choices, we find, more from flow cytometry, or if we're looking at individual cells, because we think that as cells die, they lose markers in kind of weird ways, and so it's really kind of hard to tell what's happening. Do you have any thoughts on that, and do you feel like these are the best time points?
That's the thing that we actually kind of in the middle of the investigation of this, doing these experiments, we try to see what happened if we treat the tumors in different size, and if that affect their characteristics by that. And then if we can like pinpoint the exact size that will have the best effect, that we can use it in other research.
But also timing after the treatment. Do you find that any of the particular time points are more reliable in terms of having?
The first the time that comes first. This is the time that you see the most effect of the PFAS.
Okay.
So I just have one other question. So you see that there are differences in specific pathways between the two tumor types, though there's an overall trend, you would say. Do you have plans of looking at other tumors?
Well, right now we're planning on looking at glioma tumors and to see what is the proteomics expression.
But you don't see any, you haven't yet really, nothing jumps out at you as a specific pathway that's been...
From the RNA-seq? No.
No.
It's great. It's a lot of data, and we're going to have a very deep dive to this, to analyze all of this data.
Question over here?
Do you have any idea of what fraction of cells, if any, are killed, or what the viability looks like at these different time points?
Not at this kind of experiments.
Well, we do know DNA damage, so
yeah, DNA damage.
Wait for the next talk. Okay.
Yeah, DNA damage we do. Yeah. Okay. So.
All right. Let's thank our speaker again.
So to normalize data for a natural history study actually is a really important point, because if you actually decide on a different time point to normalize to, that heat map looks completely different.
Right.
And the degree of cytokines, chemokines, and trophic factors coming up and down at different time points in the tumor growth is going to be really important, and make it very difficult in some regards, because probably the FACS analysis of the immune cell populations in there are also going to be different. And therefore, it's going to be difficult to start to compare a small tumor to, say, sonic hitting, or at least not completely ablating, but at least sonic hitting a large tumor. And so that's something we're going to have to come to grips with. What size tumors are we going to want to look at, and what population of tumors are we going to have to control for that?
Yeah, and the reason that I asked the question was we find, and particularly after thermal ablation, but just in general, that if we're looking even 48 hours out, that as cells are dying to such a large degree that on flow we see very strange looking cell populations. And so it is just because they're losing markers in very unpredictable ways.
Wait till the next talk.
Cool. All right. Thank you.
Thank you very much.
Okay, so with that said,